Today we discuss about vectors. Vectors is the most important topic in biotechnology. In this session, we discuss about list of topics. Okay. Definition, types of vectors, plasmids, applications, bacteriophages, cosmids, plasmids, shuttle vectors, and applications. Vector definition. Vectors are the DNA molecules which can carry a foreign DNA fragment to be cloned. They are self-replicating in an appropriate host cell. Let's get into the details of definition. Vectors are the DNA molecule which can carry a foreign DNA fragment to be cloned. Okay, vectors is just like a vehicle. It is a DNA molecule used as a vehicle to artificially carry foreign genetic material into another cell. Another cell means it is a bacterial species. Okay, next. Types of vectors. Okay, first one is plasmids, bacteriophages. Next, cosmids, plasmids. Wait. Plasmid definition. Plasmids are extrachromosomal, double-stranded, circular, self-replicating DNA molecules. Almost all the bacteria have plasmids containing low copy number or high copy number. Okay. Low copy number means 1 to 4 per cells. High copy number, that is called 10 to 100 per cell. The size of plasmids varies from 1 to 500 kilobase pairs. Usually, plasmids contribute to about 0.5 to 5.0% of total DNA of bacteria. Example, Streptomyces bacterial species. Look at here, this is plasmid. Plasmid are the, one moment. Plasmid are extrachromosomal, double-stranded, circular, self-replicating DNA molecules. Circular means it is the round-shaped plasmid. Okay. Double-stranded means 2 strands. Extrachromosomal DNA means either inside or outside the nucleus of cells. Okay. Next, this is target gene. Target gene means your interest gene. Okay. This is antibiotic resistant gene. Antibiotics means penicillin, kanamycin, etc. Okay. This is promoter to drive target gene expression. Means is the process by which the information encoded in a gene is used to direct the assembly of protein molecules. The cells reads the sequence of the gene in groups of 3 bases. What is the group of 3 bases? Answer is pyrimidines. Now you tell me different types of pyrimidines. There are three types of pyrimidines. Okay. First one is cytosine. Next one is thymine. Third one is uracil. Okay. Plasmid definition is very, very important. Next. Plasmids. A plasmid is naturally occurring extrachromosomal double-stranded DNA, circular DNA. Okay, this is circular. It replicates autonomously within bacterial cell. Plasmid carries an origin of replication. This is origin of replication. Plasmid vectors are the simplest cloning vectors. It is most widely used for gene cloning methods. Characteristics of plasmid vector. It contains oricite. Oricite means origin of replication process. Next, it contains selective markers such as antibiotic resistance, blue-white screening, antibiotic resistant genes. Okay. Next, plasmids are low copy number, high copy number. Low copy number means 1 to 4 per cell. High copy number means 10 to 100 per cell. This is very, very important point. Okay. It contains multiple cloning sites, easily isolated from the host cell. Host cell means bacterial species. Next, types of plasmids. Let me read through different types of plasmids. Okay. Fertility plasmid, resistance plasmid, fertility plasmids or F plasmid, resistance plasmids or R plasmid. Next, third one is call plasmid, fourth one degradative plasmid, fifth one virulence plasmids. Next, conjugative plasmid, non-conjugative plasmid, stringent plasmid, relaxed plasmid. Okay. First one, fertility plasmid, F plasmid. Carry the fertility genes. Fertility genes means transgenes. Transgenes means transfer genes. Okay. Carry the fertility genes for conjugation process, the transfer of genetic information between two cells. Two cells means two bacterial cells. Okay. Next. Resistance plasmid or plasmid contains genes that can build resistance to antibiotics or poisons. Antibiotic resistant genes means ampicillin, kanamycin, tetracycline. Okay. Next, call plasmid contain genes that encode for the antibacterial polypeptides called bacteriocins, a protein that kills other strains of bacteria. The call proteins of E. coli are encoded by proteins such as call E1. Next, fertility plasmid. First one is fertility plasmid. Second one is resistance plasmid. Third one is call plasmid. Okay. Already 3 plasmids done. Okay. Next. Next, degradative plasmid. Degradative plasmids helps the host bacterium to digest compounds that are not commonly found in nature, such as camphor, xylene, toluene, and salicylic acid. Okay, these plasmids contain genes for special enzymes that break down specific compounds. Degradative plasmids are conjugative plasmids. Degradative plasmids are conjugative plasmids. Okay. Next, virulence plasmid. When a virulence plasmid is inside a bacterium, it turns the bacterium into a pathogen, which is an agent of disease. Bacteria that cause disease can be easily spread and replicated among affected individuals. The bacterium E. coli has several virulence plasmids. E. coli is found naturally in the human gut and in other animals, but certain strains of E. coli can cause severe diarrhea and vomiting diseases. Okay, Salmonella enterica is another bacterium that contains virulence plasmid. Next, conjugative plasmids. Next, non-conjugative plasmids. Okay. Conjugative plasmids. If they carry a set of Trojans that facilitate bacterial conjugation process, and non-conjugative plasmids, it is the do not possess such genes. Conjugative plasmids, genes transfer into the recipient nucleotide. Okay. The non-conjugative plasmids do not possess into the recipient nucleotide. Don't transfer into the genes. Non-conjugative plasmids. Conjugative plasmids carry a genes into the recipient nucleotide. Okay, non-conjugative plasmid don't transfer into the genes. Okay, it is very important. Next one. Next. Stringent plasmid, relaxed plasmids. This is stringent plasmids. These plasmids exist in small numbers, that is less than 100 copies per cell. Stringent plasmid is under the control of bacterial genome for replication and segregation. Generally, conjugative plasmids are mostly stringent plasmids. Okay, next, relaxed plasmids. These plasmids exist in large number, that is greater than 100 copies per cell. Okay, relaxed plasmid is not under the control of bacterial genome for replication and segregation. Generally, relaxed plasmids are of low molecular weight and most of them are of the non-conjugative type. Okay, stringent plasmids is conjugative plasmids. Okay, relaxed plasmids is non-conjugative plasmids. Stringent plasmids less than 100 copies per cell. Relaxed plasmid greater than 100 copies per cell. Okay. This is the stringent plasmids is small number. Relaxed plasmids is large number. Okay. Next. First one is fertility plasmids or F plasmid. Okay. Next one is resistance plasmid or R plasmids. Okay. Next, third one is Col plasmids. Next, degradative plasmids. Fifth one is virulence plasmids. Next, conjugative plasmid, non-conjugative plasmid, stringent plasmid, relaxed plasmid. Don't confuse. This is resistance plasmid. This is relaxed plasmid. Okay. This is conjugative plasmid. This is non-conjugative plasmid. Next. Next, types of plasmids by their functions. Okay, most important plasmids PBr 322 and PUC plasmid. This is very important plasmids. Okay, first one is PBr 322 plasmid. Look at here. This is PBr 322 plasmid. PBr 322 is a plasmid and was one of the first widely used E. coli cloning vectors. Okay, created in 1977 in the laboratory of Herbert Boyer at the University of California, San Francisco. It was named after Francisco Bolivar Zapata, the postdoctoral researcher, and Raymond L. Rodriguez. The P stands for plasmid and B R for Bolivar. R means Rodriguez. B means Bolivar. This is scientist name. Okay. PBR 322 is 4,361 base pairs in length. Okay. And has two antibiotic resistant genes. Next, I will explain. Okay. First, you listen. The gene encoding the ampicillin resistance AMP or protein and the gene TET encoding the tetracycline resistant protein. Okay, it contains the origin of replication of PMB1 and the ROP gene. Okay, ROP genes means proteins, which encodes a restrictor of plasmid copy number. The plasmid has unique restriction sites. Okay, for more than 40 restriction engines. 11 of these 40 sites lie within the TET origin. There are two sites for restriction enzymes, that is HIND3 clavin within the promoter. Okay, of the TET origin. There are 6 key restriction sites inside the ampicillin resistant gene. The circular sequence is numbered such that 0 is the middle of the unique ECO-R1 site and the count increases through the TET origin. The AMP or ampicillin resistant gene is penicillin beta lactamase. Promoters P1 and P3 are for the beta lactamase gene. P3 is the natural promoter and P1 is artificially created by the ligation process of two different DNA fragments to create PBR322. P2 is the same region as P1, but it is on the opposite strand and initiates transcription process in the direction of the tetracycline resistant gene. Okay. Look at here. This is PBR322 plasmid. PBr322 is a plasmid discovered by Bolivar and Rodriguez. Okay, who designed it as 322? Some plasmids is there. Okay, first one is this is PBr322. PBr322 of E. coli is the most popular and widely used plasmid vector. Okay, and appropriately recorded as the parent or grandparent of several other vectors. PBr322 has a DNA sequence of 4361 base pairs. It carries genes resistance for ampicillin resistant gene and tetracycline resistant gene that serve as markers for identification of clones carrying plasmids. The plasmid has unique recognition sites for the action of restriction endonucleases such as this is EcoR1, BAMH1, okay, PST1, PVU2. This is restriction enzymes. Okay. Look at here. This is the restriction endonucleases. Okay. Restriction endonucleases are one of the most important groups of enzymes for the manipulation of DNA. These are the bacterial enzymes that can cut, split DNA for any source at specific sites. They were first discovered in E. coli, restricting the replication of bacteriophages. By cutting the viral DNA, the host E. coli DNA is protected from cleavage by addition of methyl groups. Thus, the enzymes that restrict the viral replication are known as restriction enzymes or restriction endonucleases. Restriction endonucleases have been isolated from bacterial species and some of them are commercially available. Okay, this is EcoR1. This is BAMH1. Okay, this is the restriction enzymes isolated from the bacterial species. Okay, EcoR1 restriction enzyme. This is isolated from the Escherichia coliform bacterial species. Okay, next BAMH1. This is restriction enzyme. This is restriction enzyme. These 5 restriction enzymes. Okay, BAMH1 Bacillus ameloquaceae isolated from the restriction enzymes Bacillus ameloquaceae species. Okay, next. This is ampicillin resistant gene. This is tetracycline resistant genes. These both are antibiotic resistant genes. PBr322 has a DNA sequence of 4361 base pairs. It carries genes restriction for ampicillin tetracycline resistant genes. This is markers, also named called markers. Okay. The plasmids has unique recognition sites for the action of restriction nucleases. Okay. Next. Next one is PUC19. PUC19 is one of the series of plasmid cloning vectors created by Joachim Messing and co-workers. Okay, the PUC is derived from the classical P means plasmid, U means University of California, where early work on the plasmid series had been conducted. It is a circular double-stranded DNA and has 2686 base pairs. Okay, PUC19 is one of the most widely used vector molecule as the recombinants or the cells into which foreign DNA has been introduced can be easily distinguished from the non-recombinant plasm based on the color differences of colonies on growth media. Okay, PUC18 is similar to PUC19, but the MCS region is reversed. MCS region means multiple cloning sites. Okay, PUC19. This is PUC19 vector. Okay, this is ampicillin resistant gene. This is origin of replication. Okay, this is restriction enzymes BAM-H1, XBA1, PST1, SPH1, HIND3. These are restriction enzymes. Okay, these are restriction enzymes EcoR1, multiple markers. Okay, these are SMA1, BAM-H1, XMA1, SCC1, EcoR1. These are restriction enzymes. These are also isolated from the bacterial species. Okay. This is 5' UTR. Okay. 3' UTR means nucleotide ranging. Okay. Nucleotide ranging means untranslated regions. Okay. Polylinkers. This is polylinkers or restriction enzymes. This is ampicillin resistant gene. PUC19 components N-terminal fragment of beta galactosidase LAG-Z gene. Okay. LAG-Z genes means fragments. Okay. The MCS means multiple cloning sites. This is multiple cloning sites. Multiple cloning sites is split into the codon 6 to 7 of LAG-Z genes, providing for many restriction endonucleases enzymes. Okay, that is called HIND3, SPH1, PST1, SA2, XBAL, BAM-H1, these EcoR1, these are restriction enzymes. Okay, next beta galactosidase, this is one enzyme. The ORIS site means origin of replication process. It can be used from the plasmids. Okay, next the plasmid function. The plasmids is the introduced into a bacterial cell by a process called transformation. Okay, where it can multiply and express itself. However, due to the presence of MCS, multiple cloning sites, and several restriction sites. Restriction sites means HIND3, SPH1, PST1, BAMHR. Okay, these are restriction sites. A foreign pieces of DNA of choice can be introduced into by inserting into place in MCS region. Multiple cloning sites. Okay, the cells which have taken up the plasmid can be differentiated from cells which have not taken up the plasmid by growing it on media with ampicillin. This is antibiotic. Okay, only the cells which the plasmid containing the ampicillin resistant gene will survive. Furthermore, the transformed cells containing the plasmid with the genes of interest can be formed cells with the plasmid but without the gene of interest. Just by looking at the color of the colony they make on agar media supplemented with IPTG X girl. Okay, recombinants of white whereas non-recombinant are blue. Okay. Let me explain little more about PUC19, but it is very important. Okay, if you mix IPTG and XGAL, okay, with agar medium, the resultant outcome must be white colored colonies, but not blue color colonies. Okay, if you get white colored colonies, you can confirm recombinant plasmid is formed. Okay, it is blue color ones. It is a non-recombinant plasmids. Okay, are you understand? With this explanation, PUC19 topic is over. Okay, let me recap about total session. Vectors. Vectors, it's just like a vehicle. Okay, vectors, genes transfer from one cell to another cell. Okay, vectors are the DNA molecule which can carry a foreign DNA fragment to be cloned. They are self-replicating in a host cell. Okay, vectors it's just like a vehicle. Genes transfer from one cell to another cell. Okay, next plasmids. It is very important definition. Plasmid. Okay, plasmids are extra chromosomal double-stranded self-replication DNA molecule. It is called plasmid. Extra chromosomal double stranded means two-stranded. It is a circular double-stranded double-stranded circular self-replicating DNA molecule. Okay, it is the low copy number and high copy number. Low copy number means 1 to 4 per cell. High copy number means 10 to 100 per cell. Okay, it is 0.5 to 5 percentage of total DNA of bacteria. Example, streptomyces. Okay, this is bacterial species. Next, this is antibiotic resistant gene. This is a promoter to drive target gene expression. Okay, this is a target gene. Target gene means your interest gene. Okay. Types of plasmids. Okay. This is first one is fertility plasmid, next resistant plasmids or plasmid. Next, call plasmids, degradative plasmid, virulence plasmid. Okay, conjugative plasmid, non-conjugative plasmid, stringent plasmid, relaxed plasmid. Okay, first one is F plasmid. F plasmids means the transgenes means transfer genes transfer into the two bacterial cells. Okay, between two bacterial cells. Next, resistance plasmid cells or plasmids contain genes that can build resistant to antibiotics or poisons. Antibiotics means kanamycin, ampicillin. Okay. Next one is call plasmid. Call plasmids means contain genes that encode for the antibacterial polypeptides called bacteriocins, a protein that kills other strains of bacteria. The proteins equally are encoded by proteins such as call E1. Okay, call E1 means it is a protein. Okay, next conjugative plasmid, non-conjugative plasmid. Conjugative plasmid means genes transfer into the another cell. Non-conjugative means non-conjugative plasmids means do not possess into the another bacterial species. Okay, next one is degradative plasmid. Degradative plasmids helps the host bacterium to digest compounds, that is camphor, xylene, toluene, and salicylic acid. Okay, this is degradative plasmid. Degradative plasmids or conjugative plasmids. Next one is virulence plasmid. Virulence plasmid is inside a bacteria. Okay, it is Escherichia coli from bacteria. It is the virulence plasmids. Okay, E. coli is found naturally in the human gut and in another animals. Okay, but certain strains of E. coli can cause severe uh, diarrhea and vomiting diseases. Okay, next stringent plasmid. Stringent plasmids is the small numbers, that is less than 100 copies per cell. Okay, stringent plasmid is under the control of bacterial genome for replication and segregation process. Stringent plasmid is non-conjugative. It is the conjugative plasmids. Mostly stringent plasmids are conjugative plasmids. Okay, relaxed plasmids. This is relaxed plasmids. It is the large number, that is greater than 100 copies per cell. Relaxed plasmid. Look at here. This is less than 100 copies per cell. Relaxed plasmid greater than 100 copies per cell. Okay, this is conjugative plasmid. This is non-conjugative type plasmids. Okay, it is a small numbers. Okay, this is large numbers. Next, PBR 322 plasmid. Okay, PBR 322 is plasmid discovered by Bolivar and Rodriguez. Okay, PBR 322 of E. coli is the most popular and widely used plasmid vector. DNA sequence PBR322 has a DNA sequence of 4361 base pairs. Okay, next, this is ampicillin resistant gene and tetracycline resistant gene. Both are antibiotic resistant gene. Okay, next, EcoR1, BAMH1, SAL2, PST1, PVU2. These are restriction sites. These are isolated from the bacterial species. Okay, it is a cut or split genes. Restriction endonucleases are one of the most important group of enzymes for the manipulation of DNA. Okay, these are the bacterial enzymes that can cut or split DNA at specific sites. Okay, restriction endonucleases have been isolated from bacterial species. Some of them are commercially available. Okay, this is origin of replication. Okay, or is it? Okay. After cutting and sealing process, it can be used ligase enzymes, that is called ligation process, to DNA fragments to create PBR322. Okay, next, this is PUC 19 vector. Look at here. This is also double stranded extra chromosomal circular plasmid. Okay, P means plasmid, U means University of California. It is a 2686 base pairs DNA strands. Okay, PUC vector, it is molecules as the recombinants. Okay, which foreign DNA has been introduced can be easily into the recombinant plasmids. Okay, it is the ampicillin resistant gene. Okay, this is multiple markers or or restriction sites. Okay, restriction nuclease enzymes. Okay, it can be isolated from the bacterial species. BAM-H1, X, BA1, PST1, SPH1, HIND3, multiple markers. Okay, this is PUC 19. It is a very important. PBR 322 also very important. Okay, this is 5' UTR, this is 3' UTR. Okay, UTR means nucleotide ranging, untranslated regions. Okay, this is linkers, restriction enzymes. Okay. This is also extra chromosomal, double stranded, circular, self-replicating DNA molecule. Okay, this is origin of replication or a site. these components add into the plasmids. Okay, that is lactate, protein genes. Okay, next restriction endonuclease enzymes HIND3, SPH1, PST1, SAL, SA2, XBAL, BAMH1. Okay, these are EcoR1 restriction enzymes. It can be added into the plasmids. Okay, ampicillin resistant gene, AMPR gene. Okay, these are add into the plasmid. Okay, this plasmid is introduced into a bacterial cell, that is called transformation method. Okay, after multiplying, gene expressions will be started. Okay, if you mix into the IPTG XGAL, it is a one type of a chemical into the agar medium. Okay, next the result outcome must be white color colonies, but not blue colored ones. Okay, if you get white colored colonies, you can confirm recombinant plasmid is formed. Okay, if you come non blue color colonies, it is non recombinant plasmids. Okay, it is very important. Okay, any doubts you can write here. Okay.